Okay, good afternoon. Thank you everyone for joining me as I take you on of the journey that we've been on over the last 7 or so years developing this bee breeding system and where we are today. We must feed 9 to 10 billion people by the year 2050. It is perhaps the greatest challenge that we will face, or humanity will face. Not only do we have to improve wheat yield by 60% to meet this demand, but we also must contend with rapidly evolving pathogens and climate change. An example of rapidly evolving pathogens is UG99, rapidly evolving stem rust race that was born from out of Africa. It threatens global food security. Climate change, it's predicted that temperatures will increase by 6 degrees by 2050, meaning that the wheat crops around the world are going to be subject to more variable conditions. Drought conditions more prone, heat stress events more likely. When put like that, it sounds a bit like a Bruce Willis movie, doesn't it? So how do we meet these demands? It's clear that plant breeders need to respond faster to the changing environment. Now, there's a bit of a problem with wheat breeding. It's a slow, long and drawn-out process. You can see here, this is a depressed wheat breeder waiting for his plants to grow. It can take 12 or more years to transfer new genes into adapted material, plus another 10 years or so to transfer those genes into varieties that are suitable for farmers to grow. So that includes combining those new genes for maybe disease resistance, for instance, with the necessary traits needed for large-scale production. So agronomic traits like plant height, maturity, straw strength, and obviously quality, why we grow the grain, particularly in Australia, for bread, noodles, and of course yield. So we must develop more efficient ways of developing cultivars. So it brings me to this bee breeding innovation. Here you can see the big image of the bee breeding, one of the glasshouses we have here at UQ, fitted out with this bee breeding system using the extended light photoperiods to accelerate plant development. Using this system, we can achieve up to 7 plant generations of wheat per year. Now, these concepts were born right here at UQ about 10 years ago with my colleagues here at UQ, Ian DeLacy and Mark Dieters. Now, it was really stimulated by the research that NASA was doing in order to try to grow a food supply in long space missions. So they were using these extended photoperiods to grow wheat quickly. And so colleagues at UQ, Mark Dieters and Ian DeLacy, they thought, well, why not adapt this concepts to plant breeding and speed up the plant breeding process? So today we have this bee breeding innovation, and here it is. We routinely use it for our wheat and barley research and pre-breeding efforts. And we employ it whenever possible. You can see here, that's the Minister of Ag and some other important people here at UQ coming to check out this bee breeding system. So it certainly got their attention. You might recognise this guy here. He is the most famous wheat breeder in the world that ever lived. His name is Norman Borlaug, responsible for the Green Revolution. Now, this apparently was his favourite equation, the equation of genetic gain. So if we look at the components of genetic gain, we basically want to increase all the factors on the top of the equation. So selection intensity, selection accuracy, genetic variance. We want to increase these things to some extent, genetic variance. On the bottom of the equation, we want to reduce this, the years per cycle. So, and this is where speed breeding fits in. It reduces the years per cycle. Now, Norman Borlaug introduced shuttle breeding to Simmet, where he could conduct selection and breeding in 2 different locations in the year, so to do 2 generations a year. So in theory, doubling a genetic gain. Well, now we have speed breeding, we can do 7 generations of wheat per year. So to show you where it fits in to the current development pipeline for new cereal cultivars. So we're talking about the initial phase. We're reducing the time frame. So this line development phase, the time it takes to develop new inbred lines of wheat can be 5 to 10 years. So, and this end of the development pipeline is really fixed. You can't change. You can't get out of this process. So the lines need to be evaluated and validated through thorough yield testing. So we're talking about shrinking that phase from 5 to 10 to 2 years in this speed breeding system. So very exciting, and has the potential to double the rate of genetic gain. It's simple, but has big implications. So just to give you a little bit more explanation about the how we go from seed to seed in just 6 weeks, because it's pretty amazing. This is a typical plant. There's actually a pot with 4 plants of Suntop, a wheat that's widely grown on the east coast of Australia at the moment. Essentially we get to enthesis about 4 to 5 weeks after sowing in the system. And as I said, we harvest about 2 weeks after enthesis. And so this is a plant that's about 11 days after enthesis. And so you can see the developing grain is very close to being harvested and sampled. So it's 11 days, another few days under the speed breeding system. These grains just fill a little bit more, and we get very good germination. So if we employ a proper germination treatment. Now, when we actually dry down the grain in a dehydrator for about 5 days, and the grain turns from green to brown. And the grain is not the same size as field-grown grain. It's a little smaller and a little shriveled. But when we imbibe the grain overnight, place it in the refrigerator for 5 days, and take it out back to room temperature, we get 90 to 95% germination. So no need for embryo rescue. It's a bit like magic. So yes, we can go fast. But like I said in the video, the real power comes into play when we start to select for important traits. So let's revisit Norman Borlaug's favourite equation again. So if we start to employ selection in this speed breeding system for desirable traits, we're going to be affecting this part of the genetic gain equation. So if we're selection intensity and selection accuracy, right? So we first started developing phenotypic screening methods for some of these traits back in my honours year with Mark Dieters. We worked on grain dormancy. Now we picked on grain dormancy because it's a trait that no varieties in Australia has. It means that all varieties are susceptible to pre-harvest sprouting at time of maturity. People have been working on this trait for 20 or 30 years in Australia with 0 success. So another reason why to pick on this trait. Now, what we did is we evaluated a number of wheat varieties that don't have dormancy, and a couple of elite donor lines that do have dormancy in the system, and compared it to the field expression. And we found really good differentiation. So much so that we applied it to segregating populations, and we found that we could shift gene frequency for grain dormancy in segregating populations of wheat. So very successful. So this is part of and I guess why people have had trouble with this trait in the past is because it is a complex trait. It's governed by many genes. It's also influenced, the expression of the trait is influenced by the environment. So weather conditions, temperature, and it's very hard to sample spikes in the field, very labour intensive to go out to the field and sample them right at the right time at maturity and do the germination testing. It's much more convenient to do it in the speed breeding system and walk in, and sample the spikes whenever you want to, and it removes a lot of the environmental variation. So it was a great success. So after developing the system for grain dormancy, we thought, well, why don't we try to develop some other phenotypic screening methods for other traits? So we picked on rust pathogens. Rust pathogens are highly evolving pathogens. It's proven that seedling resistance to rust pathogens, particularly for stripe rust, has been ineffective in controlling this disease. So the focus, breeders prefer adult plant resistance. So it's a more complex resistance type. It's expressed at adult plant growth stages. It provides multiple genetic barriers as opposed to the typical seedling, gene-for-gene, hypersensitive resistance that you might be familiar with. So we developed the system for all three rusts in wheat, both by themselves, individually, but also an integrated system where we could screen for all three diseases. We also developed an assay for barley leaf rust. And so, to give you an idea, this is the high-throughput triple rust screening method that we developed. So it enables simultaneous selection for adult plant resistance to the three rust diseases in the same plant generation. And it's high throughput. We can screen up to 1,000 plants per square metre. And it only requires 6 weeks to complete. And we can do it all year round. It's not influenced by the environment. We put rust nurseries out in the field, and expression's influenced by the weather conditions. Sometimes a rust nursery in a particular year, if it's dry, will completely fail. This system is completely controlled. And so not only do we have the ability to do this high throughput screening, but we can also select onto, cross onto the selected plants in that same plant generation, which is really cool because it means that we can take advantage of those unique gene combinations that we've identified and cross them to varieties or intercross them or do whatever. So very unique system. Here, just to give you an idea, this is the expression of stem rust that we have in the system on elongated stems. In the triple rust screening method, we actually are infecting different parts of the plant with the different rust diseases. So on the third and fourth leaf, stripe rust is inoculated, and we assess the plants for adult plant resistance there. Leaf rust is inoculated onto the flag and flag -1, and stem rust onto the elongated stems. So they're being evaluated on parts of the plant where they should be evaluated. And of course, in any assay, we also include disease standards, so we're able to monitor what's going on in the assay and check for interactions and make sure things are behaving like they should be. So we further develop the system for other traits that are important. Crown rot. Crown rot is caused by fusarium, and it's increasing with minimum or zero tillage practices around the world. So it's becoming more and more important. All cultivars lack adequate levels of resistance to crown rot. And that's a good reason to go and develop a system to rapidly select for it, right? So Mark Dieters at UQ has a big project where he's pyramiding multiple sources of genetic resistance in wheat for crown rot, and he's using the system in the speed breeding to select and combine these genes. We've also developed the system for barley, crown rot, and when we select for adult plant resistance in the system, we are actually imposing conditions that are similar to the field. So we will apply the temperatures and water supply and the timing of the inoculum to get symptoms and conditions that are reflective of the real world. And that's what's worked for us in the past for the rust diseases. So manipulating the temperature and conditions is critical to getting expression that is similar to the field. Yellow spot, another important disease, probably the most important disease in Australia for wheat crops. And again, most cultivars in Australia have inadequate levels of resistance. So big problem. We have one of my PhD students, Eric, is developed the screening methodology for adult plant resistance and some very interesting results comparing seedling and adult plant expression, and they seem to be different mechanisms at play. he's aiming to introduce novel resistance genes from land races into the modern wheats using the speed breeding system and the screening methodology that he's developed. Now, we've talked mainly about biotic traits with the exception of grain dormancy, but this is some new exciting work that we're doing on root traits for drought adaptation. Now, this work has been done, work has been done around the world across the cereal crops to demonstrate the importance of root traits in terms of access to water at different times in the season. Now, work has been done to show that plants with a narrow root system have the tendency to produce more roots deeper down in the soil profile, which is important for access to water during grain filling, right? Particularly under terminal drought conditions. So we developed a system where we could phenotype root angle, and root number also controls branching of roots deeper down in the soil profile. Now, before we started this work, the method for measuring these traits were very labour intensive. We had very big chamber systems, smaller gel systems that you had to fill up with agar, and they always got contaminated, and we could only work with about 10 genotypes at a time. This system, using the transparent ANOVA pot system like this with imaging, we can evaluate 24 plants in one pot and at a density of 600 plants per square metre. So that's about 10,000 plants in one room in 1 day. Oh, sorry, in 1 week. Not that fast. So very exciting. We're using this for selection, but also mapping the genes that are controlling these very important traits in wheat and barley. Grain quality. This is something we're playing with. To me, it feels like the next step with the whole speed breeding system. We're looking at protein expression in the grain under the speed breeding system and how it actually is related to field grown grain. So, and what are the components of that? So we're running some experiments at the moment investigating this in order to develop a strategy which enables us to select for protein, a very important component driving the end-use quality for bread making and noodle making. In fact, one of my French intern with me, Audrey, is actually running this experiment. Rapid gene transfer. So we have this fabulous generation advanced speed breeding technology. We can go fast. We've got screening methods for very important traits. Now, we also have high-throughput DNA markers. The cost of genotyping has dramatically come down in recent years. We can now genotype 94 samples for $5,000 and get 40,000 SNPs with known sequence across the genome. So we have these technologies, and so together with Mark Dieters during my PhD, we thought, why not exploit these technologies and combine them in a way for a purpose, for a purpose of transferring genes quickly in the speed breeding system and pick on a few target traits. So we did that. We come up with a strategy which involved complex crosses. It involved the high-throughput phenotypic screening. We employed the speed breeding generation advance wherever possible. We employed the use of the high-throughput DNA markers to select for genetic diversity along the way. And we developed two sets of introgression lines in 2 different varieties. One suited to the East Coast, which is called H45, and one for the West Coast called Wild Catch'em. Now, both of these varieties lack adequate levels of rust resistance, so we inserted or selected for triple rust resistance along the way. And they also lack grain dormancy for pre-harvest sprouting tolerance. So that's the other target trait we focused on. Now, the development of the lines was a great success. We ended up with 90 introgression lines into this Wild Catch'em background. And here we are, Mark and myself, posing in front of one of the best introgression lines that's growing in West Australia. These lines are now being commercialised by a breeding company over there. What's interesting is that we not only rapidly introgress these traits, but it seems like we haven't gone anywhere in terms of yield. It seems like we're in the same ballpark in terms of Wild Catch'em or if not better. Some of the lines are actually between 8 to 10% higher yielding. And that's just after 1 year of evaluation. So these are in trials again this year, and they'll be continued to be evaluated until commercial release. Again, the second variety, H45, a set of introgression lines, 90 to 95% similar to H45, but they carry the rust resistance genes and some level of grain dormancy. This is really promising stuff. Mark and I just visited Victoria and in southern New South Wales. We checked out the sites this year, and they look fabulous. Last year, they were up to 25% higher yielding than H45, some of the lines. Again, this needs to be validated, but very promising results in terms of we want the target traits, but we also want yield at the end of the day. It's the most important thing. Now, in any selection program, breeding program, you can think of the selection as a funnel, right? So you end up with, you start off with lots of genotypes, and you end up with a very highly selected set, small set, that has the potential to be commercialized. Now, often through this selection pipeline in a normal program, or a typical program, the selection would occur in different generations, different years, for different traits. Now, what I'm about to show you is an example of the type of selection work or pipeline that we've conducted recently here at UQ using the speed breeding system. And interestingly, this is done in one plant generation, not across years or across generations, right? So this really demonstrates the power of what we can do. So we could start with 40,000 seeds in a cross. We can select for grain dormancy. We transplant the dormant grains into the speed breeding system. We can inoculate with yellow spot. We can select the resistant ones. So we just pull out the susceptible ones. We then inoculate with the triple rust grain. We select for leaf rust, stem rust, and stripe rust. We throw out the susceptible ones. We then reselect for important agronomic traits that drive adaptation, so plant height, maturity. And at the end of the day, we end up with a highly selected set, a small number of individuals, but they have a very special gene combination, don't they? Because they've come through this funnel, and they have been enriched for all these desirable traits. So, and it's of our opinion that we should probably genotype the selected ones rather than the unselected ones, right? So they're the ones with the value. So the highly selected ones that are left over at the end, we can do various things. We can take them through to line development. Depending on the traits that we're working with, we can repeat the selection process to fix genes. We can do back crossing. We can top cross, intercross to stack genes, whatever the purpose. So a lot of this technology we developed in wheat. And we thought, why not apply this technology to another crop species? There's a lot of potential. So in 2010, as part of my PhD research, I actually travelled to Argentina with Greg Platts, a pathologist here in Queensland, with DAF. And we toured around farms in Argentina, and particularly a lot of barley is grown in the Buenos Aires province. They don't grow much wheat. And that variety, the variety that dominates in the region, the Buenos Aires province, is grown across 80% of the region, is this variety called Scarlet. Not Scarlett Johansson, Scarlet the barley. And so this variety is well adapted. It's preferred for malting and brewing. That's why they grow it. But the problem is that it's susceptible to every disease known to mankind. So we thought, well, what a great case study, right? We can take our rapid trait introgression technology and we can pick on scarlet and we can insert multiple disease resistance really quickly. So that's exactly what we did. Now, before I show you the results, to give you an idea of, okay, if we adopted more of a standard plant breeding strategy for putting disease resistance into scarlet, we could take a back cross approach. And this graph here displays the frequency of lines that would carry different numbers of donor genes, right, in that configuration. Now, what you can see is that this strategy works very well for a few genes. Okay, you can get 5%, for instance, you can get 5% of the lines in a back cross-derived population that's unselected. 5% of them will have the three or four genes. Okay, now that's going to work well when we're working with, or the target is one trait, one disease, because often we do need about 3 genes to have adequate or stable levels of resistance across environments and pathotypes. Now, what if we consider 3 traits? 12 genes. Well, that strategy is just not practical, and enough to make your head hurt. So, how do we get up here when it's not feasible? So we developed a strategy in this bee breeding system using the high throughput phenotypic screening methods for these particular important diseases of barley. Now, this is the strategy we followed. It could have been optimized a little more. We've learned a lot, and we didn't have a lot of resources doing this. We had no resources doing this. We had to do this on the weekends, right, and stuff. So we took 4 elite donor lines from the Queensland program, crossed them to scarlet, and we developed large F2 populations subject to multi-disease screening. We then back-crossed the progeny, and then generated, again, large segregating populations which were subject to these multiple disease screening consecutive cycles so that we could fix the genes. And out of that, we actually took the resulting introgression lines, right? So we employed speed breeding wherever possible. And in fact, average generation time was about 10 weeks. So when we select for these traits, we have to apply conditions that are suitable for the pathogen development and expression similar to the field. And that slows us down. So we can't go from seed to seed in 6 weeks. We actually do it in about 10 weeks if we apply selection for these traits. Okay, but that's still pretty good. So the high throughput disease screening involved screening those segregating populations, about 5,000 plants per screen, and about 1,200 per cross. We were dealing with 4 crosses. The sequential screening of the plants involved application of those 4 diseases sequentially on the different leaves as they grow. So first we applied former net blotch using 3 pathotypes that are common, representing the common pathotypes in Australia. 1 pathotype of spot former net blotch went on next, followed by spot blotch, and then leaf rust. So, plus we got an incursion of powdery mildew in the glasshouse, so we were able to reselect for powdery mildew. What a bonus! After this process, we selected between 120 and 150 survivors per screen, and each trait we applied a selection intensity of about 30%, and it worked out quite well. So the resulting introgression lines, we developed about 87 of them, and we did this in 2 years. So we did well, we did it quickly, and then what we wanted to do then is validate the levels of resistance in these lines that we developed entirely in the speed breeding system. So here they are. We assessed them in 6 nurseries in Australia, in Warwick and Toowoomba, and here's one of the nurseries in Uruguay. We're working with our collaborators there. So we're working with the pathotypes that are common in South America. So this is the results. So you'll see, again, the black line. This is the unselected back cross-derived population. In theory, the number of lines in a number of genes in the lines in an unselected population. Okay, dropping to 0 very quickly. You'll see that the red bars are actually the number of lines that carry these large numbers of genes. So we ended up with 6% of the lines that we developed actually carried more than 10 genes. And so, and we were fortunate enough to actually get 1 line that carried 11 genes. So that was pretty impressive. Now, this population size was 87 introgression lines. Alternatively, if you wanted to combine the 11 genes in an unselected backcross derived population, you would have to evaluate close to 20,000 lines for a 99% chance to combine these genes. I know what I'd rather do. I'd rather evaluate 87. So that really demonstrates the power of this technology, combining large numbers of genes very quickly in 2 years. When you think, if we had to combine 11 genes across three breeding cycles, how long that would take? So we applied the technology wheat, barley, and it actually works in other crops too. So the peanut company in Australia actually runs their whole program routinely now through the speed breeding system. So they get an extra generation a year. And they can go from seed to seed in about 8 weeks. We've actually just finished some experiments in canola, demonstrating that it's possible in canola in just 8 weeks. 8-week generation time in canola. I'd be pretty excited if I was a canola breeder right now. Sorghum, we're evaluating it here at UQ in collaboration with Ian Godwin and David Jordan. So has a lot of potential, but a lot of challenges to come over. We feel it might be possible, but it's just a matter of plant growth manipulation, right? So here's Adnan, one of my PhD students. He's been managing some of the speed breeding experiments in canola. And a lot of the Australian cultivars go through very quickly. That's days to enthesis. And we can harvest viable seed about 3 weeks post-enthesis. 2 weeks is not enough. It actually needs 3 for canola. But some of the winter types from Europe actually are delayed, and so appear to have a vernalisation requirement, similar to maybe winter wheat in the system. So at the end of the day, this technology has tremendous implications for rapidly improving the existing cereal cultivars that farmers grow. So we can take variety X that farmer Bob grows, and we can insert desirable traits that make the production better for him, right? Like, as we've done for wheat and barley. We can use this technology to rapidly pyramid genes in top crosses or traits. So the breeding companies, I would suspect, would be very excited about this, because it means that before release of a variety, you actually have the opportunity to combine very high-yielding elite lines and actually bring together complementary traits. And maybe it's an extra 2 years of development, but you end up with something very superior at the end of the day. And here at UQ, we're also using the technology to rapidly introduce genetic diversity into modern wheats. So that's a big problem. Modern wheat breeding over the past 100 years has created a bottleneck in diversity. So we're working with a set of land races from the Vavilov collection Institute in St. Petersburg, and we're actually using the speed breeding system to rapidly transfer desirable genes into Australian cultivars. And so it's all very exciting, but how do we get here? How do we have this fantastic technology that's applicable to so many crops and has so much potential for lifting genetic gain? Well, I have to say, it wasn't smooth sailing. So if we go back to 2007, in 2007, we had no funding, right? We had to do things on the sniff of an oily rag, right? And that's typical, I guess, when you have a crazy idea. It takes a while for people to come around and to support you. It included, this is me on a Sunday morning. No, not really. But I did create the first inoculation cabinet out of pieces of rubbish from the tip bin here at UQ. So no one would help. We were branded as crazy scientists with this crazy idea of speed breeding, this NASA-inspired stuff. How is that going to work? And people told us, oh, it won't work. You're crazy. You never work. You can't do that. You can't do that? I mean, they're supposed to be scientists, Someone to have this sort of attitude, Well, I have to say that it did work, and we can do that. So it goes to show that if you do have an idea, and you can, and people say that it won't work, and you believe in it, you give it a go. And maybe it doesn't work, but maybe it does. And I think that's what science is all about. So here we are today in 2014. We have the speed breeding technology. It's now adopted by several major breeding companies in Australia for both wheat and barley. If they're not doing it in collaboration with us here at UQ, they're doing it themselves, and that's fantastic. That's our innovation having impact in the real world. So we have companies in Australia, 3 of them, that are actually commercialising our wheats that we developed right here at UQ. And they're going to be grown by farmers in a few years from now in the real world. So that's very exciting. I was invited to give a proposal to the Bill and Melinda Gates Foundation in Seattle. This technology has captured the attention of people for a very good reason. And when I actually gave a proposal to the whole ag team at the foundation, and it was a surreal feeling because in view of Bill Gates' office there on the top right-hand corner, I was thinking back about how I built my inoculation cabinet out of the rubbish bin. And that foundation's been a big support of me and connecting me with people around the world who are interested in this technology and collaborating. So it's been very good. And now we have this great tool for accelerating genetic gain. And as you know now, we use it in our day-to-day research and pre-breeding activities. So I'd like to acknowledge the following people. Again, the speed breeding and rapid trait technology was co-developed here with Mark Dieters in the audience. And also some of the original concepts were also contributed by Ian DeLacy around the speed breeding before my time. My PhD students are fabulous. I can't do any of this stuff without them. My colleagues here at UQ, collaborators, and of course GRDC for their support in my development over the years, but also for the projects that we now have. Thanks a lot.